Hi there. In today's series of videos, we'll be talking about the analysis and manipulation of RNA to understand how a gene is contributing to a cell's function and how we can engineer cells with altered properties by interfering with RNA expression. In this video, we'll talk about purifying RNA and two methods for studying the expression, northern blots, and reverse transcriptase PCR. In the previous set of lectures, we talked about DNA analysis and we learned what are the entire set of genes an organism has. Today we're going to look at the study of RNA, and we'll be using microarrays and sequencing to answer a particular question: is which RNAs are made in a particular cell. Different cells have different properties in your body even though they have the same genome. The reason they're different is because they turn different genes on and off to achieve their particular functions. And this slide makes that point a little more clear. All cells in your body have essentially an identical genome, and yet they're very different. Each cell has about 23,000 genes, and when we study RNA analysis, we're trying to understand the subset of those, 10,000, 12,000, whatever, that get expressed in that particular cell type to achieve those cells' particular goals. The breakthrough technique in the mid-90s was microarrays or DNA chips. This allowed us to query the expression of each gene simultaneously so we could really get a global view of transcription and gene expression. Now, one caveat is that measuring mRNA levels gives you an amount of a transcript, not the full activity of the gene or the protein. In one case, RNAs can be made but not active because they're being bound by other protein or other RNAs. So they're ready to go if needed, but they're not being used at the moment. Secondly, we know that proteins are really the molecular machines that do most of the work of the cell, and they can be active or inactive based on post-translational modifications or, again, lots of other factors. So while we can't guarantee that knowing the RNA level will give us insight into all the things going on in the cell at that instant, because it's such a useful technique, it's so robust and gives us a truly global picture, microarrays really were a major breakthrough in the analysis of cell activity. Nearly all the methods used to analyze RNA is going to be based upon hybridization. As you know, the ability of nucleic acid strands to bind to complementary sequences via hydrogen bonding. This will be true whether we're doing northern blots, PCR, or microarray analysis. In order to study mRNA, we need to isolate it and purify it, and this slide is an overview of that process. Remember that messenger RNAs have at their 3' end a poly-A tail. That's about 200 repeated bases of A at the end of the sequence. So using hybridization, we can hybridize that to a sequence of TTTTTTT, generally referred to as oligo-DT. And this will be the principle for the interaction, which is going to selectively retain the messenger RNA. So in this example, we start with total RNA. We'll apply it to a column in the presence of large amounts of salt. Ribosomal RNA and transfer RNA will pour right through the column. The messenger RNA will stay on the column. We'll continue to wash with more sodium chloride, purifying it, getting all the other items off the column. And finally, we'll elute with H2O to get the RNA off the column. You'll recall that the reason this is good at breaking the hybridization between oligo-DT and poly-A tails is that RNA and DNA are polyanions. Like charges repel. Without the positively charged sodium present, you have no screening counterions to electrostatically stabilize the interaction of these two polyanions. So in pure water, double-stranded nucleic acid structures dissociate. That's going to release the mRNA into a tube, a highly purified mRNA fraction into the tube. So in this slide, we'll talk about analysis of specific RNAs using northern blots. You'll recall that we talked about Southern blots for DNA analysis, a technique developed by Edward Southern. Northern is just a play on the name to indicate the same technique, but with a goal of analyzing RNA levels. So just as in Southern blots, we're going to apply our nucleic acid samples to a agarose gel. We're going to run it, and we're going to be distributing all the RNAs based on their size. Because eukaryotic cells produce such a wide variety of messenger RNAs, this will be a smooth distribution without any bands visible at the RNA level. So after the RNA has been electrophoretically separated, we will turn off the voltage, lay the gel on its side, put a nitrocellulose piece of plastic. This is a positively charged plastic, which the negatively charged RNA will be attracted to. We'll put paper towels and blotting paper on top of it, and now by capillary action we'll suck water up. This will carry RNA from the gel onto the nitrocellulose filter. So then on this filter we have an exact duplicate of the gel. We can take this filter paper, put it into a bag, have a probe sequence. It can be radioactive or chemiluminescent. The probes will bind based on hybridization to the gene of interest, and then you can see in this sample which of the 4 samples loaded on the gel express that gene. And a type of study that's done frequently is that you would load RNA from many different tissues, separated on the gel. Here this would be about 15 different tissues, and you can see that the gene of interest is expressed in retina, brain, the temporal lobe, the cerebellum, but not liver, spleen, thymus, lung. So it's obviously a gene associated with neural function. Another very useful method for RNA analysis is reverse transcriptase PCR. Now RT-PCR will not always mean reverse transcription PCR. It can also mean real-time PCR, and you'll have to tell by the context which one of these methods is being used. In this class, we will not speak about real-time PCR. We'll always talk about reverse transcription PCR. Again, the goal is to measure relative expression levels of mRNA. As before, we're going to isolate and purify mRNA. We're going to reverse transcribe mRNA into cDNA. Then we can, now that we've got DNA, we can amplify it using PCR, just like we did any fragment of DNA. And then we'll do a readout running it out on a gel. So here we have the poly A tail for mRNA. And again, we'll take advantage of hybridization with an oligo-DT primer. Now we'll use the enzyme reverse transcriptase to make a copy of cDNA. Now we have a single-stranded DNA molecule, but when we put in our 2 PCR primers, we will eventually get both strands indicated by the dark red and the pink color. These are double-stranded copies. When we have many copies, we can visualize this on a gel electrophoresis. So one point I want to make here, when we were talking about making cDNA libraries, we used an oligo-DT primer to make the first strand, and we used various tricks of either it looping around or RNase degradation to do second strand synthesis. And there we would have double-stranded copies of all genes present in the cell. Here we've added gene-specific primers so that we make the DNA molecule, and this primer is not amplifying at the oligo-DT region. It is specific, unique sequence to this gene, and so we're just going to PCR this fragment of the gene. And here's an example of RT-PCR analysis, a gene referred to as DSCAM, which is going to, with the primers, is around, I guess, around 600 base pairs. And you can see that DSCAM is expressed in the heart at 12 weeks and the brain in 12 weeks, but not later in development between 9 and 23 and 32 weeks. So this is a gene that's on early in development. And here, in general, you try to make it so that the relative intensities are meaningful. And so you could even say that it's expressed at a little higher level in brain than in heart. And that was also true at the later time points, it seems to be. So RT-PCR is very useful when you have very small amounts of material, mRNA, and you can also, depending on the number of cycles you do the PCR, you can observe very low levels of expression or high levels, so you can tailor your experiment to query a wide dynamic range of levels of expression. Because of PCR's awesome power to amplify even single molecules, this is what makes it so useful at crime scenes where only a very small amount of blood is available. One of the issues with getting reliable quantitative data from RT-PCR is because of the potential nonlinearity of amplification. The difference between really doubling 2.0 the amount of material each cycle versus amplifying it nearly double 1.95, say, each cycle is huge after 30 or 40 cycles. And unless you're careful and have some good controls, you can be really fooled about the levels of a gene. Secondly, it's not a super high throughput method in that you have to design custom primers for each gene, and conditions have to be optimized for those primers, and so people won't usually use this technique for more than a dozen or so genes at any time. Nevertheless, if you want a quick and dirty answer about a couple of genes, this is definitely the methods that's most used. Okay, in the next video we'll talk about microarrays.